the whole unit then sort of tells you where we're going with this, but not in very much detail. Inheritance, this is expanding on what we've already done, because we've done the structure of DNA, you know, to the extent of the nucleotides and the actual code that's in the bases for producing proteins and protein synthesis and that sort of thing is all, you know, part of the last unit. So we're going to need to overlap a little bit. So there'll be a little bit of recap on that. But then it's a matter of looking at how that code is inherited either from one generation of cells in your own body to the next generation of cells in your own body, or from one generation of individuals to the next generation of individuals. So, you know, it's about within a generation, but also between generations. And it leads fairly obviously into the idea of evolution. That gets fairly brief treatment, I have to say. But evolution is just part of genetics, and it's about the selection of genetic combinations that improve your chances of survival. So, you know, if you've got a good genetic mix that enables you to survive in the environment you find yourself in, then you will pass your genes on to the next generation, whereas others won't. So inheritance and evolution definitely go together. I had them separated for the last few years into different units, and it's not been ideal, but yes, they are now back together again. So hopefully this will be, you know, a fairly sensible sort of sequence of things to do. Now, before I start, there are some really good websites out there. One of them, I'm trying to remember what it was called that I was referring to last time. The one I was having the problem playing the animations from. I can't remember what it was called. Sorry? No, it wasn't the McGraw-Hill one. It was, yeah. There's one called Learn Genetics. Hang on, if I have a look actually on the. If I open up the internet, it might be there on my new screen bit, the recently used stuff. No, it's not. Okay. That's a pity. Okay, I'll have to come back to the good websites. I've probably written them down somewhere, but there's a lot of stuff out there. Well, it's Your Genome, wasn't it? yourgenome.org. Yeah. I knew if I stopped thinking about it, it would come back. So, good websites. So it's your, I don't know if it's uppercase, lowercase or what, but yourgenome.org. And another one I found called Learn Genetics. So I will check those out a bit later if I've got the time. By the end of this week, I can sort of confirm those. But they, yeah, are very up to date. There's animations, there's text, there's stories, there's examples, there's up to date stuff. So, you know, there's a lot of good things out there, and it's always worth you know, keeping at the forefront of this sort of thing. Okay, now we are pretty much going to cover all the different aspects of genetics, although we've done part of it already. And if you were to sort of look at the sort of content of genetics as a topic, the aspects that you've got, you have molecular genetics, and that really is pretty much what we've done. Well, we didn't really do a lot of detail on it because we didn't do the control side, but protein synthesis. So molecular genetics is about protein synthesis. You've got the sort of inheritance side of it that we would call Mendelian genetics. Mendelian after Gregor Mendel, who did all the original, well, they were, no, you're thinking of Charles Darwin. Charles Darwin and the finches, as the birds, yeah. No, Gregor Mendel was a monk, and he was did a lot of, it was pea plants, yeah, did a lot of breeding of pea plants. But he didn't know anything about genetics, so, you know, he wasn't really giving us a, you know, an insight into the DNA and all that. But Mendelian is the inheritance part. And then you also have population genetics. And population genetics is going to look at how the number of versions of a particular gene, what we call alleles, will vary in populations. So generation after generation, they normally stay the same unless something interferes and you get evolution taking place. But population genetics can be used to predict all sorts of things, and changes in the number of particular alleles in a population, of course, will lead to evolution. And so that one will, yeah, to evolution or not really, but that one will lead us to that. So we've sort of done the first part, you know, to some extent. We're going to look quite a lot at the second bit, and then obviously the population genetics leads us to the final bit on evolution. So where are we actually going to start with this? Well, you need to not throw away all your handouts for the biochemistry unit, right? Because you do need to be familiar with the structure of DNA. All right, so a sort of recap, review that, whoops, spell it right, we're overheating a bit today. The sort of things that would be useful, you've got, yeah, DNA structure, so that, you know, you're familiar with the, familiar with all the, you know, the coding and all that sort of thing. And then protein synthesis and mutations as well, because we'll be looking at what controls gene expression, and mutations, of course, are relevant in terms of, well, definitely in evolution, because you know, that's what drives evolution, changes in genes which are then selected or not selected. All right, now the DNA structure bit that you need is really just to have that familiarity with the idea of this sequence of bases determining the sequence of amino acids in a protein. And we have, you know, quite an elaborate sort of process to produce the proteins. But any mutation, any change in the sequence of bases in the DNA can have an impact on the protein that's produced. It may or it may not. One or two things, like I said, I did promise I would read this particular book, which I've been, it's a very readable book, it's just I haven't had time to read it properly, and I'm trying to think what it's actually called. There's a book which actually is on my list over here, The Epigenetics Revolution by Nessa Carey, and it's another book by Nessa Carey. I think it's something like The Myth of Junk DNA, and it's about the DNA that doesn't immediately code for a sequence of amino acids and where it is and what it's doing. And I never seem to find enough time to actually just sit down and read it properly. It is very readable, but I've been making an effort, as far as I've got, just to make a few little notes about things. I don't remember stuff quite as readily as I used to. But this includes stuff like, when we were doing protein synthesis, we had this idea that, you know, if you have a sequence of bases that forms a gene, then that's transcribed into messenger RNA. You get the bits spliced out, and then the exons are stuck back together again to make the bit of code that's actually then translated into a protein. But one of the interesting things that I've discovered from this book, I knew this was a fact, but not quite in terms of the quantity, is that actually 60% of genes actually code for more than one protein. in terms of the protein synthesis. The thing is, you get more of those the more sophisticated an organism is as well. There aren't very big differences between us and, you know, quite simple organisms in terms of the number of bases, the length of the DNA, perhaps that sort of thing. But the more sophisticated an organism becomes, the more flexibility there is in the genome. So that you have, for example, I'm trying to think what the word was that was used actually to demonstrate this, but you know, you can have, oh, I know, it was, yes, departing, I think. If you had a gene where the exons, let's put some introns in here. Oh, I can't remember how she did it now. Not even 9. 1, 2, 3, 4, 5, 6, 7. Let's make it longer. Well, I've got 9 exons. I think I've got 10 now. 1, 2. Oh, let's see. If they coded, yeah. Let's just take one off the end, actually. For letters in a word, rather than amino acids in a protein, that one gene, you would say, right, that's the gene that codes for the word departing. Once we've cut out all the introns, which are these bits, all right, and we decode the rest of it, then we can make the word departing. But in fact, you can make a lot of other proteins from that gene. You could, for example, make the protein part, or depart, or ear, or dear, or all those different proteins could come from that one gene. And this is something that we find more and more. And this is the sort of thing that is now being discovered. All right, that genes are much more flexible than we thought they were. We used to refer to something called the central dogma. All right, now dogma is like a rule, I suppose, if you like. And it always used to be one gene, one polypeptide. That was it, you know, that a gene was the code for producing a single polypeptide. We now know it's not. And actually, you can take one gene, but you could actually code several different proteins from it. And it's how that is controlled that is, you know, is really where biological science is moving ahead in genetics anyway. So, you know, you've got quite a lot of sequences possible from that, and it looks like about 60% of our genes have that capacity to produce different proteins, because they were really quite surprised when they finished decoding the human genome. It's some time ago now, isn't it? It's about around the millennium. Can't think exactly when, but they were expecting to find something in the order of about 100,000 genes, I think it was. That was the sort of prediction, and they've ended up finding only 20,000. So what that suggests is then that those 20,000 are more flexible. They are not just producing single proteins, they're actually producing a variety of different proteins. So 20,000 genes in our DNA, each one possibly producing multiple proteins, and those 20,000 genes only make up 2% of the length of the DNA. All right, now human cells that have a nucleus have about 2 meters of DNA, but actually only about 4 centimeters of that is the coding part that we're familiar with, the genes. And it's what the rest does that is the fascinating bit. And this is what we used to call junk DNA. And there's quite a lot of scope for considering some of these things in presentations. You know, I really would recommend sort of having a look at some of this stuff and possibly, you know, finding some of the sort of more up-to-date sort of little areas to look into. Although the risk with that, of course, is that there's not yet enough there to actually make a presentation. So we have, in humans then, we have approximately 2 meters of DNA per, well, can't say per cell, let's say per nucleus, because red blood cells, of course, don't have a nucleus, so they're not going to have any DNA in theirs because they haven't got one. 2% of DNA is what we call genes. All right, so it's the other 98% that is, was known as junk, and it's that as well that varies as you get more sophisticated organisms. They have more junk DNA. We can still call it junk DNA because then we know what it means. Non-coding, but even that's a bit misleading because some of it does. There's about 20,000 genes, and you don't get very much variation between species in terms of the number of genes. Just because we think that we're the most advanced and sophisticated organisms on Earth doesn't mean we've got any more chromosomes, any more genes, any more DNA. And then we have this business that about 60% of genes code for multiple proteins. And I suppose it's worth saying the other 98% is what we used to refer to as junk DNA. And there are multiple uses for this. And I will mention a couple of them very shortly when we start talking about chromosome structure. So far from being junk, it's, lights are being shone into the junk DNA. A bit like turning the light on in my attic at home and looking around and thinking, no, none of that's useful at all. And then you start looking and think, oh, yes, it is. I'm not throwing it out after all. So, yeah, okay. Well, let's start with a bit on chromosome structure. Although we've done DNA structure, and we have briefly mentioned that DNA is wrapped around histone proteins to form chromosomes, we haven't really looked at chromosome structure in detail. And so that's going to be my starting point, really. Now, we will probably find that there's a PowerPoint involved in this, I think, right near the beginning. Ah, dear. Yeah, apologies for all the blank sheets. Okay. All right, what we have is DNA plus histones. Histones are proteins, and they come as little, they're like little spheres of, well, there are 8. They come in little groups of 8, and the DNA is wrapped around them. I'm not sure if we've actually got a diagram of that anywhere. Somewhere we might have. But I think, actually, that's probably back in... I might need to move some bits around with these PowerPoints, because things that were in the DNA structure, of course, for the last unit do overlap with this. And let's have a look. I just want to go into there. Okay, that's what I'm looking for. Bear with me, I might have actually moved that into here as well. Yeah, let's start with this. I keep getting this. Oh yeah, see which one this one is. Okay, chromosomes, as far as human cells are concerned, and that's going to be our example, we have in body cells, our chromosomes are arranged in pairs. Each pair consists of a maternal and paternal. And our body cells contain 23 pairs of chromosomes. Now, those 23 pairs are all different from each other. Well, there are 23 different individual chromosomes which each have a pair. 22 of the pairs are known as autosomes. And the 23rd pair are the sex chromosomes. All right. And so if you were to take one of your blood cells, let's say a white blood cell, because a red one wouldn't be a lot of use, but if you look at one of your white blood cells, you'd have to catch it about to divide. That's the only time you see chromosomes, but more of that Wednesday probably. The pairs that we've got are known as our diploid number. 23 is our haploid number, but that comes from the cells that we call gametes, that's eggs and sperm. Where you have 23 single chromosomes. I just need to get some of the terminology out here before I can start actually describing them. This is the problem I've got. All right, so 23 is our haploid number. And 46 is then what we call diploid. So doubled, if you like. It's rather odd. Those words, they almost look like double and half, and you never have a single sort of word in there. Just trying to think, is that enough to sort of get me started on this? Now, if you choose to do a genetic disorder for your presentation, you will need to track down which chromosome it's on, whereabouts on that chromosome it is, what sort of change in the genetic code has occurred, how that affects proteins that are produced from that gene, and so on. Interestingly, there's quite a number of genetic disorders which are not within the actual coding part of the genes, but there are disorders that relate to some of the junk areas as well, because that controls the expression of genes. And yeah, it is becoming a much more complex subject, I must say. Right, let's have a look at this. Just wondering if I've got the slides in the right order here. I think what I want to do is to start. No, I said no good. I'm going to have to do it in the order it comes. Now, this, I haven't shown you this before, have I, with the shoes? Or did I show you the shoes? You didn't see it, you didn't have my shoes. Right. Well, this goes back to a few years ago when I'd been thinking for ages. I often refer to chromosomes as being like pairs of shoes. You know which ones go together. They're very definite pairs. So, you know, a right sandal will go with a left sandal, a right trainer will go with a left trainer, a right boot goes with a left boot, and so on. And it's like that with chromosomes in our body cells. You know, a maternal chromosome number 1 goes with a paternal chromosome number 1. They've got the same genes on them. Maternal chromosome 20 goes with a paternal chromosome 20. They've got the same genes on them, and so on. And I thought it'd be really quite useful to sort of illustrate this using shoes. And I thought, you know, but you'd need 23 pairs of shoes, wouldn't you? And one summer I was tidying out the bottom of my wardrobe, and I thought, oh, well, while I'm at it, let's just see what I've got and I think 35 pairs of shoes later I realised I wasn't going to have a problem illustrating this. It's funny looking back at this now because a lot of these have gone. Yeah, I have a slight shoe habit, I think. But the nucleus in a cell that's just going about its everyday business has 23 pairs of chromosomes, but you can't see them. They are all just tangled up like a bowl of spaghetti. They are unwound. They are loose DNA, which is being opened up and transcribed and then closed up again. Another bit opened up and transcribed and closed again. So you can't see chromosomes just in an ordinary cell. You have to encourage cells to divide, and then when they are dividing, their chromosomes become organized so that they can pair up and you can get a set going to each new cell. But this was the idea, was to try and illustrate it with shoes. So this was my 23 pairs of chromosomes, or 23 pairs of footwear, and yes, there are Crocs in there. Very useful on holiday. But you can't make out much in the way of pairs there. I did try and mix them up so that the pairs weren't terribly evident. Just about see a pair of trainers, and oh, the Crocs are evident, aren't they? And the short brown boots, and yeah. But other than that, you can't actually see clearly that there are pairs there. But you know, that's how DNA would be in the nucleus when the cell is just going about its everyday business. However, when it starts to divide, the chromosomes replicate, which I couldn't do with my shoes. I couldn't get a second pair of every one. I did have 2 pairs of some. And each chromosome starts to look like this. Each one of those X's is a single chromosome, but it's been copied. Because when you create new cells, each new cell's got to have a complete copy of the DNA. So here, for example, this and this are identical. Now, the first sheet in your handouts, you've got a diagram showing a normal human karyotype. Karyotype just means all the chromosomes. And those are the chromosomes, all they're human ones, just photographed and then essentially sort of cut out and stuck together in pairs. And people used to have to do that by hand, but of course computers can do it now because they can spot patterns. But you've got 2 of each chromosome, so that is literally 23 pairs of chromosomes. What sex is that one? Male, yeah, because it's got an X and a Y at the end. All right, but more to come on that topic. The one underneath shows you what happens just as the cell's about to multiply. Each one of those chromosomes in the top image has doubled up. All right, it's copied itself. But the 2 copies are held together. There's that little crossover bit. And this is evident in all of these, this little area here. It's an area called a centromere. And that counts as junk DNA. But it's actually got a very, very specific job. All right? So there is this little area where there are very specific proteins associated with that area, and the 2 copies of the chromosome are held together. So that's still 23 pairs of chromosomes. All right? But we'll come back to the terminology on that in a bit. And if you're going to actually lay them out and number them, then they are literally organized by size more than anything else. All right, so the biggest pair of chromosomes is chromosome number 1. The smallest is 22, and the sex chromosomes are just separate. But once you've organized them by size, you then, I think, organize them by, I think it's where the centromere comes, where the crossover bit comes. So if they're the same length, then there's a way of doing it like that. But yeah, if we were going to get a full set of chromosomes, you know, you could literally take a pair of scissors to that diagram, cut them out, line them up, and you've got your karyotype. With the shoes, oh, this was a nightmare. This was on my landing. Why I did it on the landing, I have no idea, because I then had to photograph them from above. So I think I had a chair right near the top of the stairs, which wasn't a good move, really. But, you know, it's this idea of organising them in size and then in groups that had similarities. So I would love to say these are the only shoes I had, but, you know, like I say, I only needed 23 pairs and I found I'd got 35, so some got left out. But, you know, it started with the long boots, the shorter boots. So the boots were together, and then the sort of heavy shoes, and then a bunch of trainers, and then sort of work shoes, high sandals, and then sloppy shoes, flat sandals, and then I counted the Crocs as the sex chromosomes because they're different. But you could see they're grouped together, and very often it's not so evident in that top diagram, but you can see it is in here. You've got those three, then those two, then a bunch of 7 across there, 3, another 3, 2, and a 3 there. So they, you know, they're grouped by similarity. Now, when that's actually not a very good image, and it's funny, they often look, no, it doesn't even look so good close up now. Those are actually the replicated chromosomes, and what you would have on here, each one of these, there's 2 bits. Oh no, the ink's not working, as it did tell me that actually, I remember now. Get back to it. Each one of these, you've got 2 bits sticking up and 2 bits sticking down, held together by a little join in the middle there. And so that's our 23 chromosomes, or 23 pairs of chromosomes, that we've actually got, you know, there's 23 pairs already, that's 46, but if each one has copied itself, we've actually got 92 strands of DNA there. Now, each of those strands is called a chromatid, just to make, you know, to add another term, but each chromosome becomes a pair of chromatids. So each pair of chromosomes becomes a pair of pairs of chromatids. So yeah, we end up needing some other words for this. You can see that there's a banding pattern on these, and one of the things that we're doing in this unit now that I haven't done before later on is to talk about using gene probes and to talk about how you find certain genes in all of that. You know, where's the gene for producing insulin, let's say, you know, in all of that, where is it? And it is possible because we can reverse engineer. We can start with a protein, and if we know the sequence of amino acids in a protein, of course, we know the sequence of bases we're looking for, don't we? Potentially. And what you can do is produce in the lab a short piece of single-stranded DNA with those bases in that are the reverse code you're looking for, and you can label that with radioactivity or fluorescent colour, so you can actually then put it in with the chromosomes, and it'll actually just sort of light up the area where that particular gene is. We'll come back to that. But you can do it with the whole set as well, and that's what this next one shows, that you can actually fluorescently label chromosomes, which I think is, yeah, if you haven't got anything better to do, it's a good way of spending an afternoon, I'm sure. And there's a process called FISH, which is fluorescence in situ hybridization, which is actually getting active DNA to take up fluorescent labels and labeling it all within cells as well. So, you know, we can do an awful lot with this information. We can track down specific genes in all of this. Now, I think we get back to the shoes. Yeah, all right. If these are 2, I haven't got either of those anymore. If this is 2 pairs of chromosomes, then we know, for example, that this one does not go with that one, this one doesn't go with that one. And we haven't got 2 of these or 2 of those. We know we've got a left and a right of each. And they have certain similarities that tell us that that is a pair. And usually we manage to put a pair on, don't we? You know, my husband at one point possessed, I think it was 3 pairs of sort of brown trainers, and yeah, I caught him in Asda one morning with a mismatched pair. And I had, can't remember which sandals, they appear on here somewhere, which came in a brown pair and a black pair, and I swung my legs out of the car one morning, looked down at my feet and found, yeah, brown on one foot, black on the other. That was awful, ruined my day, that did. But normally, No, normally, you know, we would recognise, wouldn't we, that you know, the left and right go together, and we know that they've got the same features. You know, these are made up of 3 strands on each side. They're tied together in the middle. These have got closed toes. Those have got open toes. You know, we know which shoes match. It's the same with the chromosomes. They've got the same genes. In a pair, you will always find the same genes. Now, before the cell divides, you have to copy that information. And these were the 2 pairs of footwear that I had, 2 pairs of each. I had 2 pairs because they fit really nicely and they were very comfortable, which is not easy with my feet. I have exceptionally wide feet and it's really hard to find shoes that fit. So what this represents is the fact that we had 1 pair of black slip-ons, but now they've copied themselves. So we've got 2 left ones. No, we haven't. We've got 2 right ones and 2 left ones. And the same with the sandals. 2 right ones, 2 left ones. All right, so they are not, they don't go around then as a pair of left and right, but you get them, and I should have tied them together with a bit of string or something to show that, you know, when chromosomes replicate is the word, and that's hopefully this afternoon. I just want to get on to DNA replication. That they, you know, the 2 bits would be inseparable until the cell's dividing. So in a chromosome, each individual shoe here would be known as a chromatid. All right, so you have 2 lefts and 2 rights in each pair, or 2 maternal and 2 paternal. But we don't call them chromosomes at that point, we call them chromatids. All right, looking a little more sort of sensibly perhaps at actual chromosomes, I think you've got some of these now. I was not going to give you the slides that had all the shoes on. That's just silly. Right, what we've got here is a very, very straightforward illustration of some of the parts. When the cell starts to get ready to divide, you would start to see chromosomes, but actually you will usually see them already duplicated. It's not until they finish dividing that you then start to see the single chromosomes again. Now, hopefully next week you'll actually be looking at chromosomes in cells that are dividing. This is very hit and miss. We can never predict what's going to happen. But you can do it in root cells, and it's in the very, very small roots that you get. And I remember a long time ago doing it with, I think it was broad beans or runner beans or something like that we used to use. You'd soak them and then wrap them up in a piece of wet paper towel in a jar so light could get to them, and they would start to produce little shoots and roots. But over time we were finding that was less and less effective, so we sort of started looking for other sources of roots. And one of the things that works really well is garlic. And if you take a clove of garlic, what we do, I say we, what Jill will do, take a clove of garlic, I think you stick a pin through it and then sit its bottom end into a test tube of water, and it will start producing little tiny roots. If you've ever grown your own garlic, that's how you do it, isn't it? You know, you take the bulb, take it apart, and you know it will grow. But we have to be careful that the roots don't grow too long. They need to be actively producing new cells, but not maturing. And it's a really tricky thing, just getting them to the right length. So we might be lucky, we might not. We'll just have to see what happens. What we tend to do is to put them in sort of several days in succession to sort of see if we can get some that are just right. And you cut off the very, very tip of the root, no more than ½ centimetre. And you don't use the cells right at the tip because they're just pushing through the soil. But just behind that, you have cells that should be dividing. Any further back into the root, and they're just elongating. But those cells that are dividing, they don't all do it at once. And you might find out of a slide that's got several thousand cells on it, you find one dividing cell. But when you do see it, it is, you know, it is really impressive, to be honest. And I'm hopeful that, you know, we might find several. Sometimes you'll get one slide that's got a dozen on it. Yeah, we will see what happens. You don't see the chromosomes moving because the cells have been killed. We've sort of frozen them in, you know, in mid-movement. But what you will see are just stained sort of strands positioned in the cell, rather than just the nucleus that you've seen in previous cell samples. So some of the terminology here then. Well, the thing on the left here is a chromosome. The thing on the right is also a chromosome, but this is after it's duplicated or replicated, where it now consists of 2 chromatids. And we describe the chromatids as sisters. Don't know why, sisters rather than brothers. But on the right, you've got 2 sister chromatids. It's a slightly misleading term because sisters are not necessarily identical, whereas chromatids are. So it should be perhaps twin sister chromatid, identical twin sister chromatid, if you're going to be precise. But that representation on the right shows what's happened when the DNA has been perfectly copied. Well, we assume it's perfectly copied, of course things could go wrong. And the 2 copies, the 2 sister chromatids, are held together by this bit of junk DNA or non-coding DNA, the centromere. So, you know, the fact that some DNA doesn't produce proteins, well, yeah, no, it has other jobs. Along the length of a chromosome, you have sections of code that we know as genes. An average human chromosome, well, if there are 20,000 genes and we've got 23 pairs of chromosomes, that means we're talking about roughly 1,000 genes per chromosome, aren't we? Ish. All right, so the, you know, the middle-sized ones, you could expect to have probably about 1,000 genes. And we will find that there are different forms of these genes, but that's another story when we get onto the genetics bit. So at the moment, what we're doing is just copying a complete set of the genes on each chromosome, so that when this particular cell, when it divides, then the left-hand chromatid here can go to one of the new cells and the right-hand chromatid can go to another. So you're giving each of the new cells a perfect set of chromosomes, a perfect copy of, you know, the original. If you look specifically at a chromosome, that's a rather odd choice of diagram, I must say. If you look at a chromatid or a single chromosome, the terminology is the same. It doesn't make any difference. There are some other useful words on here, or useful bits of terminology. You will usually find that either side of the centromere, you've got one piece that's slightly shorter than the other, or very much shorter than the other. So we have what we call a long arm and a short arm. The short arm is given the letter P and the long arm is given the letter Q for identifying the position of genes. So, for example, I don't know the position of any genes particularly, but you could say, oh, the position of this gene is 11p 36.4. So it's chromosome 11, P says it's the short arm, and 36.4 is how far from the centromere it is. I think you count outwards from the centromere. So you've got, well, it's a location, sometimes called a locus, and it's a bit like putting a postcode into a sat-nav. You know, it enables you to find something. So every gene has a location on a chromosome, identified by the chromosome number, which arm, and then how far from the centromere. Now, the other useful thing on here, we'll say a little bit more about, well, sort of tempted to now, is the telomere up at the top end of that side over there. The telomere is another piece of junk DNA, and I'll write a little bit about them when we're talking about DNA replication. But the telomere is like, well, it's like an aglet. What is an aglet? Anybody wearing shoes that lace up today? It's the thing on the end of a shoelace. There you go. There's a useful word. All right. It's yeah, an aglet is A-G-L-E-T, is that little piece of plastic at the end of a shoelace that stops it fraying and coming unravelled. And that's what a telomere is, a bit like an aglet. All right. So why is it there? Well, it stops the chromosome fraying. It's not quite as simple as that, but telomeres, and I really got quite interested in the telomeres when I was reading this. This Nessa Carey book I'm reading is really good. I think I need to get a hard copy of it. I think it's probably easier to pick up and read in bits than the Kindle version, to be honest. But the idea of a telomere is that it protects the ends of the chromosome. And the problem is, when you copy chromosomes like this, the replication of the DNA can't go quite to the end every time. So that means every time you copy the DNA, it gets slightly shorter. So chromosomes actually shrink slightly each time you copy them. All right? And there is a limit to that. Short telomeres are associated with aging, and there are certain genetic conditions where the telomeres are excessively short, and that causes aging. In the reproductive cells, we have an enzyme called telomerase, which actually rebuilds the telomeres so that babies have full-length telomeres. But the older we get and the more our cells divide, the shorter the telomeres get, to a point where the cells then can't replicate anymore. They stop dividing, which is why elderly people have problems with healing. You know, if they cut themselves, their skin doesn't heal very well. Their immune systems are not so good because their white blood cells can't replicate so well, because the telomeres are shorter, so the cells can't always divide. And I suddenly realised it's an argument against exfoliating your skin. Because we always say, oh yeah, if you exfoliate your skin, it encourages all these new cells to come from below. So what we're doing is encouraging our skin to age, actually, aren't we? Because the cells can only divide a limited number of times. It'd be interesting to sort of follow, wouldn't it? You know, do people who exfoliate actually show aging skin earlier? Because you're limited as to the number of times the cells can divide, because the telomeres are getting shorter each time. Telomeres are also interesting because certain cancers are characterized by the fact that they have an active telomerase and they can keep rebuilding their telomeres, so they become potentially immortal. And that's the problem with cancer cells, isn't it? You know, that they just divide over and over and over again. So so much of this is, potentially really sort of fascinating. I ought to be writing some ideas on the board as we go. I'll try and filter out a few of the bits before the end of the afternoon. But so the idea of a telomere, I will add that somewhere, but I think on their own they would be sort of quite interesting. So I'm just going to put them right over here. All right, so telomeres added to the potential topics on there. All right, now, if we were to look at an individual chromosome, chromosome number 1, this gives you an idea of how this numbering system works. All right, if you remember back to this, we've got the short arm is known as P, the long arm is Q. So what do we got here? I'm not sure there's that much difference between the lengths of those two arms, is there? But the part on the left here is identified as P, the short, and the right-hand side is Q, the longer. But you've got an idea here of how you might identify the position of certain genes. So, for example, 1p33, 1p21.3, 1q22, 1q24.1, 1q41. It tells you, first of all, the number at the beginning tells you which chromosome it is. The P or Q tells you whether it's the short or long arm. And then the number after that tells you how far from the centromere it is. So this little nipped in bit in the middle is the centromere. Okay, it's another area of DNA then that's not actually coding, but it's a very important part. It's not only the bit that holds the two chromatids together when they replicate, but it will attach itself to structures in the cell when the cell divides, so that it's done equally. Chromosome 1 is all very well like that. It doesn't tell you what's on there, and you won't be able to read this, and you may or may not, depending on your eyesight, be able to read the version you've got in your handouts. That, I can't remember what the website is that produces all these. Yeah, I would not stand a chance with any of those on there. But, I just had to fit it into the space I'd got. Chromosome 1 consists of 246 million base pairs being the ones matched on the two strands of the DNA. And this is just a list of some of the disorders that are found on chromosome 1. All right, these are not the normal functioning genes necessarily, but these are potential disorders on chromosome 1. So if I get close enough to it, I might be able to, I don't know that I can actually, it won't help because of the resolution of it. There's things like, there's a breast cancer one there, colon adenocarcinoma, so that's one of the genes that can contribute to bowel cancer, cataracts up at the top. Another colon cancer one on here, neuroblastoma, which is a nasty one, glycogen storage disease, there's a leukemia there. But very often it's not just going to be single genes that are responsible, they're going to be genes on other chromosomes as well. All right, so this is just a selection of the disorders that are associated with chromosome 1. There's one for Alzheimer's down there as well. And yeah, a lot of others on there. Of course, chromosome 1's the biggest, so you're going to find a lot on there compared with the others. A slightly smaller one, there we go. Well, it's at the other end of the extremes, so at least I could make this one bigger. Chromosome 22, which is the smallest of the autosomes, only has 49 million base pairs. And a selection of the disorders associated with chromosome 22. So yeah, we start with cat eye syndrome. Cat eye syndrome, it's the shape of the pupil. They're slit-shaped rather than round. And then we've got things which, I'm trying to think if there's things that will mean something. Oh, schizophrenia susceptibility locus. So schizophrenia, while it might not be genetic as such, there is a certain gene variant at that location that has an impact on people's susceptibility to it. And that's, you have to bear that in mind, that a gene isn't always just an on-off switch. What else we've got? There's another cataract one, a leukemia one. Amyotrophic lateral sclerosis, ALS, that's one form of motor neuron disease, I think. I think that one is that one, I think, is the one that's otherwise known as Lou Gehrig's disease, which Lou Gehrig was an American baseball player, and it's a form of motor neuron disease, I think. And then various others on here, polycystic kidneys, trying to find things that might actually mean so. There's an epilepsy one there, manic fringe. I get that sometimes. What, manic fringe? Yeah. Well, mania is it's a mental health state, isn't it? You know, manic fringe will mean people perhaps who are prone to mania. And mania is one of your conditions in bipolar. You get the depressed and the manic. Yeah. So you know, it's amazing how how broad it is. Drosophila. Drosophila is a fruit fly. Okay. All right. Well, that might not be what we think then. Okay. Well, Drosophila is the standard organism that you do genetics practicals on. Remember, breeding fruit flies for months on end. What else have we got down here that might mean something? There's a deafness gene there, a colorectal cancer. There's an autism one, and it sometimes, well, most years, somebody will say to me, "Can I do autism?" And I always say, only if you can find enough genuine material to make a presentation, because there are genetic links, but it's not clear, it's not inherited directly. You know, it's not a one gene, one condition sort of one to do. So yeah. So anyway, that's the other extreme. There's still a lot, you know. So it's hard to get away without anything, I guess. But a lot of these, presumably, you know, we never really know about. Hopefully. No, blue people.
Yeah.
Are they blue?
Yeah.
All right.
I don't think they exist anymore, but they were quite inbred.
Yeah.
Actually supposed.
Oh, right. Well, yeah, I mean, you will get, I mean, one of the things I'll talk about is if you've got a small population, that you will tend to get what they call genetic drift. So you'll either you lose certain genes or alleles, and you'll gain others where, you'll find that, yes, if they're in one particular area, all of a sudden you get, you know, a high proportion of people showing it. But no, I've never heard of that one. Then we've got these two. The X chromosome and the Y chromosome. Now, obviously, there's quite a considerable difference between these. The X and the Y are our sex chromosomes. If we've got two X's, we are female, and an X and a Y would be male. Yeah, men definitely have a lot. Men have a lot missing. And in fact, on that Y chromosome, it's only one gene that causes maleness. And I'll talk about sex chromosomes. It fascinates me, that the way that the Y chromosome works is fascinating. But there is, must be on there. Oh, that bit. The sex determining region Y, the SRY gene. That's it. That's the only important bit. Now, if that bit doesn't function properly, then an individual won't become male. Now, there is a woman in the news at the moment who is a model who has just announced that she is intersex. Between sexes. She has an X and a Y, but appears to be female. And I cannot remember her name, but I only noticed it on my Google News yesterday. Yeah? No, I can look it up in a bit. But what's happened in that person? You shouldn't really say she, but I think she's happy to be she. Is that she has an X and a Y, but that bit hasn't functioned properly, and her body hasn't responded in a way that makes her develop into a male. So although she's got the male chromosomes, her body is female, because female is the default. Now, that's an issue when you come to, you know, who was created first, man or woman. It'd have to be woman, wouldn't it? Because woman's the default. All right? Man's only a version of that. Oh dear, we didn't do the best job we might have, perhaps. But the thing is, in men, the X chromosome is very, very important because that little Y chromosome doesn't carry very many genes compared with that huge X. So anything that's on a man's X chromosome is likely to be expressed. In a woman, on the other hand, she has two X chromosomes, and they are so big and so dominant that, in fact, in females, one of the X chromosomes has to switch off in every cell. You can't, otherwise they argue too much. But yeah, you only need one functioning X chromosome. So in women's cells, and it's random as to which one turns off. Oh, this has some really, really interesting implications. All right, because particular conditions that are carried on the X chromosome, you know, it depends which one you've got that's functioning as to what will actually happen. And it's one of the reasons that you can get significant differences, particularly between female identical twins. Because you've got identical twins, they've got the same sex chromosomes, but you might find that in one twin, all the maternal X chromosomes switch off, and in the other twin, all the paternal ones, and they could look totally, you know, there could be huge differences between them. But in terms of genetic disorders as well, it's, yeah, it is really very interesting. So we'll be doing sex linkage and sex inheritance of sex and that sort of thing as a separate bit. But yeah, there's a big difference between those. Right, that little bit, that's it. As far as male development's concerned, that's it. So yeah, it is fascinating that that one little area of that one chromosome can be so important. If it doesn't function properly, then it defaults to female. But the other thing is, it can actually get translocated. Do you remember when we were talking about mutations? Translocation was an option. And that can actually, because it's near the end, it can actually be translocated onto another chromosome and function there. So there's some weird stuff going on. And it really fascinates me. And I think if I was going to go off to university now and do another degree, can't have any more, I would possibly do genetics. I think 3. Well, I did my first degree when I left school. I just did biology. Well, it started off joint honours biology and chemistry, and then I dropped the chemistry. So I've got 1 degree in biology. Then I did an ordinary bachelor's degree with the Open University, really just for interest and to broaden the things I could teach. And then I did the master's degree with the Open University as well, and that was mostly medical science. So the other one with the OU was things like maths and technology and stuff, and a bit of computing and those sort of things. But yeah, I'd go and do genetics now because I think it's absolutely fascinating. Right, let's move on a little then. A chromosome then is not, you know, a simple thing necessarily, but you know, there's definite bits of terminology that we can use to identify the parts, and some of these bits we're going to come back to. Now, I want to do the bit on replication of DNA, but this is about, well, it's the beginning part of cell division, really. So I'm going to start up. There are 2 types of cell division, depending on what sort of cells we need to produce as an end product. If we are going for growth and repair and replacement, and we want to produce more cells exactly like the parent, for example, you know, if we're just constantly rubbing cells off the skin, we need to replace them from below. We just want to make more skin cells. If, on the other hand, we want to make egg cells or sperm cells, we have to do something different. So we have 2 types of division. We have mitosis. All right, and this involves a single division that produces 2 identical cells. Well, the cells are identical to each other and the parent cell. All right, so this one is for growth and repair. The other sort for producing reproductive cells or gametes is called meiosis. Now, those two words are painfully similar. All right, I can never really find a way of helping people remember which one's which, apart from mitosis has got the word toe in it. All right, so think body parts like a toe. If you want to make more toes, you need mitosis. And meiosis has got an E in it, and it's E for eggs. That's the best I can do, I'm sorry. This time, we actually have two divisions, and we have the genetic material producing new combinations as well. So new combinations of DNA, I will be explaining how, but we get a process called crossing over in this, and we get two divisions. To produce theoretically 4 haploid cells. All different. Haploid means they have a single set of chromosomes. I didn't say haploid or diploid for the mitosis because it could be either. It depends what you start with. You can take a haploid cell, put it through mitosis, you get 2 haploid cells. You can start with a diploid cell, put it through mitosis, you get 2 diploid cells. But with meiosis, you always get these haploid cells with a single set of chromosomes. They're all different from each other, and this is gamete production. All right, gametes are egg cells and sperm cells. And the purpose of that, of course, is to produce cells with a single set of chromosomes. So when you bring 2 together, you then restore the pairs. Now, I mentioned the words haploid and diploid. There are others as well. There is tetraploid. I'm trying to think. I can't remember what's tetraploid, but I know that modern bred wheat is hexaploid. It actually has 6 of each chromosome. And it's because of the way it's been produced over the years, the sort of inbreeding between different strains. And you end up with, 6 of each chromosome. Slightly odd. So I mean, there's something to look at if you feel like it. So if we're going to divide the cells, then we need to be able to copy the DNA. And this happens in both types of cell division. Okay, you need to be able to produce new DNA. In the first one, it's obvious, because if you're going to start with one cell and you want to produce 2 identical cells from that, well, you've got to make a copy of the DNA to go into each new cell. For the second one, it's not so obvious because we're reducing the number of chromosomes. Your first thought would be, right, if you've got one cell and you want to make gametes out of that cell, well, you could just half it and send a single chromosome to each of the new cells. And that would work. But it's the way this has evolved. And the production of these haploid cells has been a sort of evolutionary development, really. And it would have happened, mitosis would be the first sort of division that would happen. And then somewhere along the way, you'd get mitosis, and then it sort of, you'd almost get like a little hiccup, and it would do it again. It would divide again. So we still get the replication in meiosis. Once the DNA is replicated, you then get the bits of chromosomes actually exchanging with each other, so you get new combinations. Then they separate once, then they separate again. So it's it's quite a lengthy process. I did say theoretically you get 4 haploid cells. You do in sperm production, but in egg cell production you don't get 4 identical cells. The process doesn't even complete until an egg's fertilized, and you get 1 that goes ahead and may become the embryo, and the other 3 don't actually play any part. They're not all equal egg cells. But yeah, we don't have time to do a lot on that, I'm afraid. Anyway, first thing we need for both of these is replication of the DNA. Now this is described in a very specific way. It's described as being semi-conservative. Right. And what that means is that if you start with a piece of DNA, all right, there's my double-stranded piece of DNA, then what happens is that the process conserves one strand from the original and then builds a new one against it. All right, so semi-conservative means it conserves half of the original, like that. Now, when we were talking about DNA structure, we talked about the template strand and the non-template strand. And the template strand, that was fine. We could see that was a template for making proteins, for copying out a sequence that we'd translate into a sequence of amino acids. But what about that non-template strand? What was it there for? It's for this. If we start off, if this is the template, and this is our non-template, then what you get, of course, is this one will be a new non-template. Whoops, non-template. Built up against the original template strand, because we know about base pairing, which is how this all happens. And then, of course, the other one, this is a new template strand built up against the original non-template strand. And so that's the function of the non-template strand. Of course, every time this happens, you conserve half the original DNA. It's one of those things that has on occasions kept me awake at night, is that I wonder what happens to your original DNA. You think about it, you start life as a single cell with a single set of DNA, don't you? Then that's copied and you become 2 cells, then that's copied and you become 4. But those original strands of DNA from your parents are conserved. It's sort of, you think, well, I wonder if they're still in part of me now, or whether they turned into a skin cell and they disappeared. I suppose for the different chromosomes, they could end up all over the place, couldn't they? But yeah, it's just one of those things. I think, I wonder what happens there. Yeah, it must be possible to find out, because you can radioactively label the originals and then put cells through several sequences of division, and look at an embryo perhaps, and see if you can follow where that radioactivity's got to. But yeah, I haven't got that far. You know, it's like I say, it's one of those things that sometimes might wake me up in the middle of the night, but then by the time I've gone back to sleep again, I don't bother then to look it up when I wake up in the morning. You'll see that we're now coming on to another PowerPoint. It's the only way to do a lot of this, and we will have a look at, well, we'll see how far we get. We might do all right with this. Cell division up. Ooh, update 2017. This sounds promising. Right, this is the problem, isn't it? The Smart Ink thing. So this means I can't write on the PowerPoints. Never mind. I'm sure we'll manage. The links that I've put on there, I think, are rather out of date. I have no idea how long ago this started, but it's gone through several versions. So I've updated it this year. I think I've put all the PowerPoints onto Moodle. But there, you know, there's so much out there, and I've got quite a good animation, I think, associated with this at some point. It would help if I could find it now. So maybe not with this bit. This will take us through all the steps of mitosis. I don't really want to rush right through into that this afternoon. So what I want to do is to have a look at the DNA replication bit, but then leave it at that and then be able to pick up from there. But I will, yeah, I'll have a look at those two websites I suggested before we finish as well. Okay, now this I think is an image that's very similar to one that we had before when we were talking about the structure of DNA, that we start off with our double helix up there. The size of that, 2 nanometers across, so, you know, very small. One thing I was reading in the Nessa Carey book I was telling you about was that to try and get an idea of the scale of this, because this is about packing all the DNA into a nucleus, it was the equivalent of, oh, I can't remember what the thing was that she was trying to pack into trying to pack. No, it's something to do with the height of Everest and a rope. It's no good, I'll have to reread that bit. That's the trouble with reading just before you go to sleep. But it is an epic amount of condensing that has to be achieved to actually be able to get the 2 meters of DNA into a nucleus. All right. Oh, it was a rope the height of Mount Everest packed into a golf ball. That's the equivalent. It's funny. You see, I have to stop thinking too hard and then it pops into my head. But yeah, if you had a rope as high as Mount Everest and you wanted to pack it into a golf ball, you'd be looking at the same task as you've got here, is packing 2 meters of DNA into a nucleus. Now, obviously, if it was a rope and it was the thickness of a rope, then you'd be in trouble. You wouldn't stand a chance. But if you then reduce its diameter to something less than that of a human hair, then you'd be, you know, a lot better off. But even so, you know, you wouldn't just sort of scrunch it all up together into a sort of tangle. You'd have to wind it very carefully and fold it and pack it and wind it again and that sort of thing. And that's really what this is about, all right? So that's the equivalent. It's a rope the height of Mount Everest being packed into a golf ball. So the DNA, to start with, is wrapped around these proteins called histones. And they come in these little cores of 8 little histone molecules, which provide a sort of scaffold for all this. But when we go a little bit further, we will touch on epigenetics, the idea of modifying aspects of our genetic composition without actually changing the genes themselves. And it is possible to add methyl groups to the DNA and also, I believe, to the it's ethyl groups to the histones, I think, or they usually say acetyl groups. And that can actually change the way in which genes are expressed. So the histones are very important for more than just this reason. And here, what they're doing is really just giving a structure to the DNA as it's packed away. So we wind it round that, we fold it a bit, we wind it some more, and we end up with a chromosome. Now, if we were to look at it in a very, very simplistic way, this is how replication produces 2 new DNA molecules from one. So our original one at the top there, original DNA molecule with a blue strand, which might be the template or non-template, it doesn't matter, and a sort of pinky purpley sort of bottom strand there, which is obviously the complementary one with its base paired sequence there. They have to be separated from each other, and then a new strand is built up against the original code using an enzyme called DNA polymerase, and consequently you end up with 2 new DNA molecules, both of which are identical to the original and to each other. They must be, if you think about base pairing. If only it was that simple, it would make life a lot easier, but this process is very complex. Now, let's work our way up to this. What we're aiming to do is to start with a pair of chromosomes, all right? This is a single pair of chromosomes here, carrying gene A, all right? Not necessarily the same form of gene A, but once the DNA has been replicated, both the blue one and the red one in that pair have become a pair of chromatids. The pairs of chromatids are held together by centromeres, and all the genetic material on the blue one has been copied, so that where you'd got an uppercase version of gene A on the original chromosome, you've now got a copied version of that, so you've got 2 of those. And similarly, where you had a lowercase version of gene A on the red one, then that's also been copied. So you get identical copies. That's all that's showing you. Okay? Right. Now this gets a little more complicated, although this is not that different from the one we just started with. We need to separate the 2 strands, and we haven't got any enzymes on this one at all, so they've obviously got to come in. We're going to need an enzyme that separates the strands. We're going to need an enzyme that joins the new nucleotides together. We're going to need a supply of nucleotides, and there's actually a lot more to it than that as well. Now, the diagram that you need to understand is this one. And this will come up in the exam, so you can make a little note by this. All right. First question in the exam is basically, what's going on here? You'll find a lot of different versions of this particular diagram as well. It was confusing me because I'd found one and then thought I'd gone back to the same one, and it wasn't. Okay, our original DNA molecule is over on the far right-hand side. The only label that it's got is a 3 and a 5 with the little dashes, which, if you remember, are 3' and 5'. And those are to do with the carbon atoms in what? In the ribose, well remembered. Or even the deoxyribose, because it's DNA. Yeah, well done. So yeah, 3' and 5', those are the carbon 3 and carbon 5 in the deoxyribose. Prime because we're trying to distinguish those from the ones in the bases. So various things happen. There is an enzyme over here. We're not too fussed about this one. Topoisomerase. Topoisomerase unwinds the helix. It sort of flattens it out, so it's a bit more like a ladder rather than a spiral. All right. Not too concerned. I don't need you particularly to know that one. However, as we move along, this blue enzyme here is called helicase, all right? The DNA helix is, it's separated into 2 strands by helicase. At least that's got a, you know, reasonable name there. Actually, majority of enzymes have. So, you know, if we were looking at a sequence here, the first important thing is that helicase separates the two strands. So it breaks those hydrogen bonds between the C and the G, between the A and the T. So the two strands separate from each other. Now, the little purple blobs on that slide that say single strand binding proteins, not too fussed about them either. They just happen to be on there. If I'd got functioning ink on here, which I still haven't, have I? No. It's a little bit annoying. I would probably put in brackets the topoisomerase and the single strand binding proteins. Those binding proteins are really sort of like a bit of scaffolding that just holds the strands away from each other. Because otherwise, once you've separated them, yeah, what's to stop them just coming back together again? So, yeah, they're a bit of support. But the only important thing so far, really, for us, is this helicase. Okay, now what's going on? Well, we've got two strands of our original DNA. And they're clearly not being copied in quite the same way. But the reason for that is that there is one enzyme available to do this, DNA polymerase. And the problem is that DNA polymerase, obviously, being an enzyme, it's got a stereospecific active site. It can actually only work in one direction. All right? It can work from, and I never remember, I rely on the diagram to tell me. Right, here's the original 3. DNA polymerase works from 3 to 5. All right? It won't go from 5 to 3. So it's making quite a neat job along here, isn't it? So they call this one the leading strand. This isn't a problem. The DNA polymerase sits on here. We get new nucleotides coming in. We haven't got those on the diagram. It would clutter things up a bit, I think. And the DNA polymerase manages to move along this strand, the 3 to 5 one. It just goes along here fine. So that no issues. The other strand, of course, goes from 5 to 3. And DNA polymerase can only go from 3 to 5. So it has to go backwards in chunks that then have to be joined together. So it can only copy from right to left on this diagram. So we're going to get mistakes in this, I'm sure, all the time, but that is what happens. What we have to have is a whole bunch of other stuff going on here. You can see, for a start, there's an enzyme called DNA primase. Not too concerned with that, but it will join on a little single, there's actually likely to be more than that, a little RNA primer, a little starting point. It will sort of find a bit of that strand where the copying can start, and it'll attach a little bit to it, a little bit of complementary, a few complementary nucleotides, so that you've got a starting point, and then it will have to be copied in that direction. Okay, and then the same thing would happen here, and you'd get a primer attached there, and then it would copy in that direction, right to left. You've then, of course, got to join it all up. Now, these little individual bits are called Okazaki fragments. That's not particularly difficult. The name, you know, is obviously you just got to remember the name or recognise the name, depending on how I write the exam. But these are the little chunks that have been created in a backwards direction that then have to be joined together. So we end up with another enzyme here, DNA ligase. The ligase, when we did enzymes, we had a little bit on the classification of enzymes. I did say we'll come across ligases. There's one. And it's like, if you think of the word ligament that joins bones together, a ligase joins these fragments together. All right, so there are important bits in this, really, are the helicase that separates the strands, the DNA polymerase, the DNA ligase, the Okazaki fragments. But what it means, there's another form of DNA polymerase here, which I think that's the one that checks. You have like almost like a proofreading sort of process going on as well. I lose track of it somewhere in all of this. But the diagram on the next slide is it's a much simpler version of this. But the process is not quite as straightforward as that one would have us believe. All right. Theoretically, all we're doing is building a new strand complementary to the original. Oh yeah, of course we are. Yeah, we're going to produce copies very nicely, but yeah, this is what's actually involved. You've got to flatten the strand out, separate the two pieces, then you can let one side get on with it because that's going the right way for the enzyme, but the other side has to go in these little backwards chunks, which have to be joined together. Once they've been made, then you join it together with the ligase. And so that's described as the lagging strand. Lagging means it's slower. If you lag behind, you're behind other people. So leading and lagging. So that one is copied faster than this one, not surprisingly. There's a bit more to this actually, but that I felt was quite a useful addition to this, although perhaps in colour they don't always help, to be honest. But it's the other way around. The diagram's been flipped here. But down at the bottom, we've got the leading strand that goes from the 3' down here in the bottom left. And the copying for that, the green blob on here is sort of where it's got to at any given time. Or, now the copying has got to green blob. And down at the bottom there, the leading strand has just been replicated, just in a straight line. Whereas the lagging strand had to be done in bits. So we had to go from 3 to 5, from 3 to 5, from 3 to 5. And then these little 3 sections are the Okazaki fragments that have to be joined together. And the point where the 2 strands are separated by the helicase is called a replication fork, for obvious reasons. You've got 2 bits forking away from each other. The curious thing that happens with this is that actually when DNA is being copied, it doesn't just do it from one end straight through to the other end. Okay, I can't remember how good this is, but as I've put it in, it must be reasonable. Oh, great. And I literally only put that in sort of a few weeks ago. That's a pity because I think that was quite useful. I'll have to find something else. Okay. This is what actually happens. All right, rather than it just starting at one end and making its way to the other end, you'll find it happens and you get these replication bubbles along a piece of DNA. The helicase will open it up, but then it won't actually just go one way. It'll actually be going both ways at once until, of course, they will meet up. So, yeah, a curious sort of effect there, but it can start in several places. Along a given DNA molecule, you can have several replication origins, and then each of these is a replication fork, and it will separate out and go. So which bit is lagging strand and which bit is leading strand, of course, will be different in the 2 directions. It will, won't it? For any given bubble and any it's going to be depend on which way it's going, isn't it? So yeah, the actual replication process theoretically, could take a long time, but because you get it happening in multiple places at once, it does speed up the process somewhat. Now, we're not going to do the crossword just now. We'll save that as a starter. What I want to do is to just track down those 2 websites and just check that those are what we need. Right, let's. Okay, it was your. Oh, it's not remembering it. yourgenome.org, wasn't it? Is it .org? I don't know what I've written there. Genogamy. Genome.org. Yeah, this was the one where I was having trouble playing the animations. I can't remember what it was we did about it in the end. Might have to use Chrome to actually do it. But there's an awful lot of really useful stuff in this one. DNA replication for a start. Let's try this. That's not promising, is it? Look. Okay. Let's open Chrome and see if I can get it to do it with that. All this wonderful East Coast College stuff on here. Your genome. Ah, right, okay. I clearly have played around on this one. Yeah, right. The feeling that we haven't got any sound.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction.
That's
The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
That's really good. The 5 to 3, of course, it goes from the 3 to 5 on the template, which means it's building the new strand from 5 to 3. was what she was referring to. So, you know, in terms of, yeah, I was thinking, did I say that wrong? But no. I didn't emphasise the RNA primer starting bit, which is worth, you know, having a look at that perhaps you know, a few more times. And then that, what was it, exonuclease that cuts out those little RNA bits and replaces them with DNA bits. That's just adding a bit more detail to it that I didn't have in there. But, you know, as far as animations are concerned, I think these are really good. They're clear, they're concise, they use the right terminology, and above all, they're not American, which, yeah, and I shouldn't say that, but to me, it makes it easier to listen to. So yeah, all right, yourgenome.org is definitely a good one to use, and just get rid of that one. The other one that no, if I go back to Google, sorry, I should have looked at a lot of other stuff on there, but that Your Genome's got loads of stuff. You can just explore through that. There are, you know, there's information, there are other animations, there's stuff that might even inspire you towards ideas for the presentation. Now the other one, Learn Genetics. I'm trying to think. I bet that's, oh, it won't be its actual, oh, it is. Oh, that's useful. Right, this one, Genetic Science Learning Centre. Actually, I think I've put links to these on Moodle. I remember doing it now. This was one that I came across just by chance. You know, and often that's the way that you come across some really good stuff, you know, without anybody saying, oh, you need to use this. And it had got quite a few things in other than just genetics. So although it's called Learn Genetics, you've got this section up here on the top left, you've got basic genetics, pigeon breeding, you know, because you all do that as a hobby, don't you? The pigeon breeding bit is sort of what's behind Charles Darwin's, a lot of Darwin's writing was talking about domestic chickens and pigeons. Variation and selection, variation, selection and time, epigenetics, that's a useful bit. Genetic science, well, let's just have a look in there. Transgenic mice, measuring gene expression, genetics in Utah. I think that's where this comes from. GM foods, farming for pharmaceuticals. Yeah, that's a strange way of putting it actually, but farming, actually, I will put that on here. That's spelt PH. While I think about it. It means using, well, as it says, transgenic technology for creating animals that make drugs in their milk. DNA profiling, that's a good option for a presentation as well. Using genetic tools to fight viral outbreaks, conservation genetics. Well, I could just, you know, copy the contents out of this over onto there, to be honest. And that's just what I've got there, Genetic Science and Society there. So it's a bit of a random choice. But you've also got other things on here, and I'm not sure, some of them are directly genetics related, others not so obviously. You've got under human health, metabolism from food to fuel. That was probably more used for the last unit actually. Precision medicine will be to do with genetics, I guess. Genetic disorders, let's have a look, see if we've got anything particular in there. Genetic screening, single gene disorders, chromosomal abnormalities, multifactorial disorders, so loads in here. Gene therapy, family health history, those are, you know, genetics related. Under the cell biology section here, we've got stem cells and cloning. Stem cells are part of what we're going to do in this unit because stem cells work in controlling development of tissues by turning certain genes on and certain genes off. So, you know, that's putting it very, very simply. Cloning, well, it can be about transferring genetic material, so that can work. Science tools, you've got virtual labs in here. DNA extraction, gel electrophoresis, PCR I'm going to talk about probably next time, because PCR is, nah, let's not do it now because, yeah, PCR is like an artificial form of replication. Sometimes you want to multiply up a sample of DNA and you only got a tiny little bit, you know, it might be from one cell that somebody's left at a crime scene and you need to multiply it and copy it. And the polymerase chain reaction is just an artificial form of replication. You have a machine basically and you put your little DNA sample in it, let it run for a while and it separates the strands and copies new ones on, separates those, copies new ones on, separates those, copies new ones on. So it just doubles and doubles and doubles and doubles your sample. So you've got, yeah, applications like that. And then there's ecological, I'm not sure about the genetics in there. Let's have a look. Microbiome change. Can't see anything immediately genetic in there. But you've got things like antibiotic resistance, which is. So there's a huge amount of stuff actually in there that, addiction, genetics in the brain. So there's a basic neuroscience bit. But that, there are more and more sites like these now where you've just got this sort of very, very well presented material that actually supports quite a lot of what we do. Okay, let's stop at that point.